afternoon, everyone. My name is Diego Nieto, and I am a research professor at UTPL in Ecuador. So I'm going to present you about the chemical and biological treatment of free cyanide from a gold mine. Here is a brief outline of what I'm going to talk about: the introduction, the objectives, methodology, and also results and discussions. I don't know if anyone has listened something about Ecuador, but Ecuador is really known for their fruits, actually bananas, and also for the production of oil. And besides that, it's also known for their all natural environmental thing that has turtles, frogs, that are really endemic in some regions, exclusively in the Galapagos Island. For example, according to the Ministry of Environment in Ecuador, we are the 17th nation within all the mega diverse countries around the world. However, right now, Ecuador is changing, according for the government, change from the production of raw materials like bananas and oil to the mineral extraction. But that mineral extraction is not well done right now. It's just done by untechnical, traditional techniques done by the population of the people living all around the Andean highlands. Actually, all the mines, gold mines in Ecuador, are located in this region and also in the Amazonian region. That is also known for the all the animals and endemic species that live over there. So the objective of this thesis was to determine the conditions for different chemicals used for the treatment of effluents containing free cyanide, and also use a rotating biological conductor and determine the hydraulic retention time to treat these effluents into different concentrations. So how we did this? We went and take some samples from a gold mine industry. As you can see here, this is a reservoir pool that contains free cyanide at 280 milligrams per liter. And we also took some samples from after one treatment, that is a chemical and physical treatment. They reduce the effluent until 10 milligrams per liter. So we took different volumes, as you can see here, 150 liters and 90 liters, from these two pools. After that, we designed an experiment for 3 different reactants, as you can see here, sodium hypochlorite, hydrogen peroxide, and carrot acid. We did a factorial design 2 to the 4th, and we ran 48 experiments and controls, and we do it in a jar test, and we analyze the free cyanide degradation. For the biological treatment, what we did, we use a microbial consortium isolated from soil contaminated with free cyanides and has the ability to form a biofilm. For that reason, we use RBC. As you can see here, what we did first is to inoculate the reactor with around 10% volume of the biofilm. We start with 300 milligrams of sodium cyanide, and also we use an enrichment medium. We let the batch culture first to run in 21 days to first start building the biofilm on the disc, and then we run two experiments in a continuous system with different fluxes to determine the correct hydraulic retention time. What we measured here was the degradation of free cyanide. We maintained the medium at 10, pH of 10, we ran the disc 16 RPMs, and the temperature was 20 degrees Celsius. As you can see here, There are a microbial consortium that is under investigation with metagenomics right now that has the ability to form biofilm early Mayer flags, as you can see here. So how works the RBC here? We have the flux coming the first stage, second, third stage, and then we have a setter in the fourth and fifth stage, and then the effluent comes out. What are the results? For sodium, first of all, the metal content of the effluents, whereas you can see, this we have copper, iron, magnesium, but actually what is interesting here is the amount of copper containing in the effluents, because it has enhanced some reactions with some reactants. I will tell you that later. In the case in sodium hypochlorite, we see that the main factor was the volume of the reactant added to the experiment in this case, followed by the time. Then for hydrogen peroxide, we see that the main factor was the volume added to the effluent. And for carous acid was the time, followed by the pH. So when we run the experiments, we had that the hydrogen peroxide removed around 92.73% of free cyanide, and 46 and 51% was due by the sodium hypochlorite and carous acid. As you can see in the bars here, number 4, there is the removal from in the green column. We can see the removal actually done by the reactant, and we have also in the control that has some free cyanide is removed by the oxidation by oxygen. As we can see in the jar test, all the atoms are rotating, so that has an effect also over the volatilization of free cyanide to the environment. So, as in some investigation, we have found that hydrogen peroxide is the best chemical used for the treatment of free cyanide, but also is enhanced by the copper ions. So if you have an amount between 10 to 20 milligrams per liter of copper ions, that will act as a catalyst and enhance the reaction and the reduction of free cyanide. In this example, we have levels of copper ions higher than 10 and 20 milligrams per liter. And also, the amount of hydrogen peroxide used in this investigation was in excess. So that helped to remove that percentage of free cyanide from the effluent. Also, we can see in these figures that the time wasn't essential for this investigation. Yes, because the time needed to degrade or to remove free cyanide was lower than we expected in these cases. For the biological treatment, we ran an ANOVA test to see which hydraulic retention time was the best. And we can see here that the best time was 10 hours. So, because we have around 85% of free cyanide removal, as we can see here, 84 is the experiment number 4, which contained an initial 280 milligrams per liter of free cyanide and 10.13 hours of retention time. Also, this was enhanced by some volatilization of the free cyanide due by the rotation of the disk in the reactor. What happened, as you can see also in experiment number 6, that the total removal was 97%. However, the biological removal was 81.62. What happened here in control, that if we maintain longer times the effluent running, we are going to have is volatilization of the free cyanide for longer times, and that will reduce the activity of the biological treatment in this case. So for that reason, we think that better the retention time will be 10.80 hours. We can see here the removal of the cyanide and also the growing of the biofilm in the reactor to 10 hours. And the comparison between the three hydraulic retention times, as we can see in the blue line, if we maintain longer the time, we will have an effect on the free cyanide removal in this case. For 280 and also for 10 milligrams per liter of cyanide. When we review these results with the literature and also with what is expected by environmental law, so we didn't get to those limits of free cyanide in this aspect. And comparing with EPA and TULAS, that is from Ecuador, both treatments didn't get to 0.2 milligrams per liter that need to be an effluent to be discharged to any river or lagoon. So, as a conclusion, we have that the best reactant was hydrogen peroxide, enhanced by the copper ions present in the effluent. The best hydraulic rotation time was 10 hours. And both treatments didn't meet the EPA and TULAS limits for free cyanide. Thank you. Have any questions?
Any questions? Yes, please.
So what is the consortium composition? Which are the organisms present in the consortium?
Excuse? Will you repeat?
You have used the consortium, no? Microbial consortium for the treatment of cyanide. Microbial consortium.
Yes. What is the?
What is the composition? Which are the organisms present in the consortium?
What is the? We are running the experiments right now. We just isolate the microbial consortium, so we are running the molecular identification of the microorganisms present in that consortium. The possibilities are pseudomonas, that gender of pseudomonas doing the work right now, and that's the results that we have in that aspect. Yeah, we are just running right now that molecular characterization.
You showed in the beginning these two different pools of water, where there was 280 in the first one and 10 mg in the second one. So that is the current method of removal of cyanide? Is that the difference between the two?
The first one is, what I'm going with this is that right now some industries are throwing away effluents from 280 and 10 milligrams per liter to the environment. In this case, this industry have just a big pool and maintain that effluent with that concentration of free cyanide, and after that they make a physical and chemical treatment. However, they don't meet after that treatment the environmental laws established by Ecuadorian government, so they discharge also the effluent containing 10 milligrams per liter.
Because from 280 to 10 milligrams, that's about 95, 6%. So what method is that?
INCO process is called, where you use you evaporate the effluent first and use another chemical to reduce the contamination with free cyanide. So in this case, we are trying to give another way or another vision, another alternative to the people that use these kind of methods also.
That's quite a lot of water you have to evaporate. That must be very expensive. Yeah.
Exactly. What happens when we use these kind of reactants? For example, hydrogen peroxide is cheaper. So what you just need to do is to apply a amount of the hydrogen peroxide and just mix it, and it will free cyanide will reduce, as you see here, less than 1 hour. Yeah.
Okay. Any more questions? No? Okay. Thank
Thank
you.
you.